Hello, this is Kaylee Hasel again presenting for Biology 2440, our cell lab, where we were giving a list of vocabulary words pertaining to cell membranes and their functions. I took this list and broke it down into two groups. We had our cell layers and our cell models. So we'll go ahead and get started with our cell layers, and then we will move on to cell models, and then I'll let you see the entire map up close so you can see the parts of it. First off, we'll start with the cell layers. The three cell layers that we were given in the vocabulary sheets were the membrane proteins, your plasma membranes, and your phospholipid bilayers. Our bigger group of words that we were given were our membrane proteins, which have several different types of proteins that create this group. You have your transporter membrane proteins, your receptor membrane proteins, your anchor membrane proteins, enzyme membrane proteins, Mons layer associated membrane proteins, lipid link associated proteins, protein attached associated proteins, integral membrane proteins, and peripheral membrane proteins. All these together make up your membrane proteins that we were given for the cell. Moving back over to your plasma membrane and your phospholipid bilayers, these contain structures called vesicles, which are little fluid cell sacs that assist in transportation of fluids in and out of the cell. The vesicles use three different types of channels to help with this movement. You have membrane channels, gated ion channels, and ion channels. When you break down the gated ion channels, you have electrochemical gradients and concentration gradients. This means that this type of movement requires different concentrations of solution both inside and outside the cell. If we come on our concentration gradient a little bit further, we were given the terms hypertonic solution, isotonic solution, and hypertonic solution. These three indicate the types of cell movement that you're going to have. If it's hypotonic outside the cell, then it's going to pull solution outside of the cell to equalize, to balance it out. If it's hypertonic and outside the cell, then the cell is going to suck in solution into the cell to balance it out. It's constantly looking for a balance between the two voids. Coming back to electrical chemical gradient and concentration gradient, when you have these two, they use four different types of transportation: osmosis, passive transport, simple diffusion, and active transport. These four are your main modes of transportation. Passive transport actually also can break down and use channel-mediated passive transport when you follow the graph back up, it goes back to your gated ion channels. We were also given two cell models to look at in this vocabulary list. We're both given the sandwich model by Davis and Donnell and the fluid mosaic model by Singer and Nicholson. These two models break down and show the actual layers of the cell as a 3D image, as a tactile kind of a form for us to study the cell with. The four words that were given with these two models were the amphithetic molecules, the detergent molecules, glycoproteins, and lipids. Detergent molecules are a little bit difficult because these are actually used to break down your lipids when they interact. Lipids will break down into three different types of fats. You have your unsaturated fatty acids, saturated fatty acids, and then our general lipids. Glycoproteins actually contain proteoglycans, and then our amphithetic molecules have two sides of the molecule. You have the hydrophilic head of the phospholipid and the hydrophilic tail of the phospholipid. I appreciate you watching, and I hope you're having a wonderful day. Let you see the concept map just a little bit closer. And then our cell model. I do appreciate watching, and thanks for your time.